nice place to start with your career. So, and this call actually including the genomic, bioinformatic, tumor modeling, pathology, therapeutic development, and the translational clinical trials. So as Scott mentioned, our group and actually working on patient-derived xenograft models, and so we have about 350 PDX models cover many kind of cancer tissue. So we are part of tumor modeling as well. So just give a little bit of history and back to about 20 years ago, actually, before we came into the picture. And there was a consent in the field. And so prostate cancer patient-derived xenografting was extremely hard. Only when tissue come from advanced metastatic tissue can occasionally grow in mice. So that was a consensus. But as I always, I was told by Jerry Cunha, any kind of statement is a conditional. When I study what's wrong with this statement, it turned out everybody just using subcutaneous grafting site, and then with an extremely low engraftment rate. So when we came into the picture, we just using liver capsule, renal capsule, prostatic capsule, and to test where you can have a higher successful rate in terms of survival. It turned out renal capsule is a beautiful grafting site to keep all sort of tissue viable, no matter is advanced cancer or even primary cancer, even pre-malignant lesion. So this is a grafting site. Actually, the reason for that is probably very simple, because the kidney is a very, very small in our whole body, but 27% of cardiac output goes to the kidney. That's why the kidney is red. And that's a place to give you really rich blood supply, particular at the critical 24 to 48 hours period. Of course, this wasn't received very well, but nonetheless, over the 20 years and this grafting site, we routinely use that. We continually prove it. And so this is just to give you an example. And in terms of prostate cancer, and this is the only list practical useful model. And the majority of them actually come from a hormonal naive prostate cancer tissue. And we can go to the detail, but we talk about that later on. And so the hormonal naive prostate cancer still the most difficulty, kind of a cancer to grow. But in our hand, that's the sweet spot, because when you have a hormonal naive prostate cancer xenograft, and then you can put them in animal, and then mimic the clinical kind of a treatment and monitor the progression. So I will emphasize why that kind of model is so useful. So to cut it short, the model in our hand is kind of not only just unique, actually we can frozen down them in early generation to avoid, kind of an unnecessary evolution and also save the cost. So we can demonstrate our collection cover all the known, molecular subtype and currently. So the model has been used for many, many studies. So we talk about that in detail later on. So currently, there are a few major activity in the lab. I just want to use them as some example to share my idea with you. So to learn from the role models from the leader, actually, we're trying to also focus on discovery all the way heading to translational or knowledge generation. So this is really focus on initial discovery, mainly use our models, and then identify and the potential target, and then eventually really enter the drug development field. And that's not necessarily only path. And the knowledge generation also one of the major, kind of task for us as for everybody else. So I want to use a few examples of how we use a model. And, the part one I want to talking about is, majority efforts really focus on, the deadly disease, either AR positive CRPC or NEPC, which is certainly necessary and critical. And, but the challenging also is if you start the intervention too late, you may not generate a big survival benefit for the patients. So the one of the focus we're trying to do, can we do something earlier and before CRPC or NEPC, and recurring and fully developed. So then that's something we have this kind of model. And the one example is we're trying to focus on NEPC transdifferentiation. And this transdifferentiation is really cool because people from time to time using different terms. Now they call this cellular plasticity, became really, really popular. And you could call cancer stem cell, you could call this and that. But fundamentally, and in order to be plastic, and the cell has to be undergoing dedifferentiation. And, the level of dedifferentiation decided how plastic they are. So the first question we ask, what are the early driver that are preparing adenocarcinoma cell for NE transdifferentiation? So this is the first phase. And then in order to do that, you have to knocking down and some lumina adenocarcinoma features, such as like AR and PSA and REST and so on and so forth. So to study this one, as Scott mentioned, and we have some unique model. This is the one we call LTL331. This is really cool model because we get from patient who demonstrate a typical adenocarcinoma. You can see the glandular structure, they are positive and was purely adenocarcinoma with a relatively well-differentiated phenotype, actually. So it's hard to establish this model. It took us 3 years to get this adenocarcinoma line going on. Then we mimic the progression by, host castration. That's one example. But surprisingly, after almost 9 months, and tumor came back, but no PSA. And then we check, we got lost. The tumor come back, PSA did not come back. So we say, is this still human cancer? Is this still prostate cancer? If so, what kind of prostate cancer it is? It doesn't express PSA. So after all those kind of, after answering those kind of questions, we are convinced. So this adenocarcinoma, somehow after host castration, after after a long time quiescent in animal, when they come back with a NEPC phenotype. So of course, that was 10 years ago. When we report this phenomenon, nobody believed it. And so we have to do the hard work. So repeat the experiment 3 times each time it took us 1 year to finish this experiment. So, but we were convinced, but that's not we were convinced at the very beginning, but nobody trusts us. But the good news that after 3 years, and the donor, the patient's tumor actually turned out NEPC, but it was almost 3 and 4 years later after we knew that. So from then, and some people, particularly like Martin Gleif, start to trust the model more before that, and that was a kind of just one type of model anyway. So when I had a nice conversation with him, I said, you see, our model predict your patient tumor progression. And but in any case, that was really sweet. And the model really showed the consistency which was seen in the patient. So then we trying to study these things to say, hey, and is this what are the major driver for this kind of transdifferentiation? So the first question we want to ask, is this a genomic contribution? And by working with Colin, and we demonstrate there was no major genomic alteration during this transdifferentiation. So then we came up with a hypothesis. We say that must be epigenetic. And then we're trying to say, okay, the epigenetic may have to come up earlier that actually help us to suppress the adenocarcinoma features, and then preparing the cell ready for redifferentiation into neuroendocrine prostate cancer. So then from there, actually we report one of the gene called HP1 alpha. And so I don't have to go into the detail. It turned out HP1 alpha really can enhance the H3K9 trimethylation. And then that histone methylation and happen at the promoter region of AR and REST and suppress the adenocarcinoma features. Of course, HP1 alpha is not only one player in that space. So the second question we want to ask, then if a cell get ready, what are the early drivers that direct the dedifferentiate adenocarcinoma cell heading for NE cell lineage redifferentiation? So that actually you have to thinking about something earlier, because most of study really focus on terminal NEPC. So no matter you find BRN2 or ASCL1 or anything else we know, those are the transcription factor only associated with the terminal well-differentiated NEPC. But we also trying to say at the early stage, and what are the NE lineage commitment transfactor that may play a key role in cell lineage transition, and maybe continually important in maintaining the NE phenotype. So that's actually very cool. So we use a bulk sequencing, follow the time series. Of course, we get a lot of ideas of what could be the transcription factor. And then, of course, and the bulk sequencing is also kind of give us some difficulty in terms of heterogeneity. So in working with Colin, we actually did the single cell sequencing, as you can see, and there are multiple subgroup of adenocarcinoma. There are multiple group of neuroendocrine, and you're using all kind of markers. You can sort of, divide them into different cluster, and then So through this kind of single cell sequencing, and we also identifying potential intermediate cluster that give you the transition middle stage. So to cut it short, actually, we found the cluster 17 likely is the early and transition phase, and then go to the cluster 10 and the cluster 1. So from here, you can see, and some of the transcription factor like PROXX1 and really start to show up, and then getting higher and higher in cluster 10 and cluster 1. And by contrast, those SOX2 and NKX2.2, ASCL1, BRN2 mainly show up in the terminal cluster. So this is the kind of efforts we're trying to focus on. And of course, we want to say is this is still continually higher in patient, and whether or not also correlated with NEPC patient sample. So as you can see, this PROXX1 and relevant to NEPC cell line, NEPC PDX, and also at a protein level, and also show good correlation in the clinical setting. So we did some functional study. It turned out really, really important when you overexpress these transcription factors. Of course, many of those neuroendocrine markers start to show up, but a marker just a marker. And when we do some functional study, and you find this knocking down is extremely effective, which is and also specific to NEPC. So I want to move on to the second part. So the second part, I want to not only focus on something earlier, I want to talking about something more important, because it's not so hard to find a cancer-associated gene. There are hundreds of thousands of them. But the question is, which one is a soldier? Which one is a general? And if we spend all our time marry a soldier, you fulfill the duty for the task as for soldier. So that may not be ideal for your long time efforts. So, and we know cancer is a very challenge, although we feel very hopeful. And also we often hear cancer is a complicated disease. It's not because we're not smart enough, because the cancer is too complicated. I think we all agree with this levels of reasoning or excuse. So that's okay. And so when one of my students and Stephen Chow joined me, and now 11 years ago, and that time the second edition hallmarker just a paper just published. So the student was so smart. I don't want to give him a project like just focus on a gene and then get a paper, get a thesis. I give him a hard task. I said, go back to the cancer biology Bible to educate yourself, to figure out what are the most fundamental features for cancer, and then decided where you should go first. The reason I challenging him that time, I feel, there were something wrong with this Bible, because one thing we know, and we predicted, the number of hallmarker would keep increase if knowledge increased. And it turned out our prediction was definitely correct. So in 2022, you got 14, which increased from 10. So, and another question we were asking is that when you put all those hallmarker into a ring, you really don't know which one more important than the others. So they are fundamental critical features for cancer must have. There are some features that nicely for cancer to have. There are some cancer enabling features. So what is really more important ones and also truly fundamental and unique to cancer? That was the question, and I don't know the answer, but that was a question to my student, and so I really forced him to have a different look at the hallmark of cancer, and so he did. He trying to say, okay, there are some kind of hallmarker that mainly associated with survival mechanism. There are some phenomena that mainly associated with cancer, aggressiveness. There are some phenomena that mainly kind of enabling the cancer to be aggressive cancer. So at least you can divide them into 3 categories. And so then I asked him to look at the whole thing. I said, okay, and most likely we get used to go to the details, always go up, and you are the number 1 in all the branches. That's actually the scientific directions. So is that the only direction we should focus on? And so we perhaps need to go up. We also need to go down as well to see what is the common feature that cancer may have. That's actually was really interesting. I want to give a credit to Donald Coffey. And I remember when I was a postdoc with Jerry Cunha, I overheard Don Coffey said one thing. He said, tumor is bizarre, and they often living in the lymph node, just like a criminal living in the police stations. And that was really interesting. That's stuck in my brain for many years. Then the first thing I asked my student was, if a criminal can living in the police station, they need to wear the uniform or speaking their language. And, if that's the true features, how are you going to describe that? He actually was very, very brave when he was young, when he was only 21 years old that time, before he was contaminated. And he came up with a hypothesis like epithelial immune cell transition means the successful cancers, epithelial cancer cells need to learn how to communicate with immune cells. So that was nobody likes the ideas. There are so many new terms, although they could be significant, and we can argue about that. But one of the major challenges we face with, there are too many those kind of immune factors that normally don't express by epithelial cell, but turn on. And just to give you example, look at the cancer cell and the T cell communication. They can turn on so many immune, kind of related genes. So this is not mentioned. They are B cells, NK cell, macrophage, and so on and so forth. So there are so many small languages for them to use. So I forced him again. So I'm sorry for that, but he was really a nice student to work with. So I said, okay, because there are so many immune genes turn on in cancer cell, and if the seems EIT is real, but what is the major communication code cancer use to influence most of the immune cells, but not only just a small language? And so that could be give us a more critical and practical kind of answer to the question we have. So he moved on to look at the hallmark of cancer. So it seems that all solid cancer hold two factors. One, the tumor microenvironment always immune suppressive. Otherwise, they will be killed. Second, the tumor microenvironment is always acidic. Everybody knows that. Everybody knows that. And that was that's really common. Now, I did some survey. I went to a few immunologists, asked them the question, is there any connections between acidic and the immune suppressive microenvironment? So the answer was mostly no. And, they know a lot of molecular mechanism in details, but they don't believe lactic acid or acidic microenvironment has an impact on anti-cancer immunity. So again, this Stephen came up with another hypothesis. His hypothesis was, okay, actually, cancer cell generate lactic acid, make the microenvironment into pathological condition. That's perhaps the major language that cancer use to suppress anti-cancer immunity. So that was a kind of really cool hypothesis, and truly not only just look at their contribution to cancer biology, but mainly related to cancer survival. And so over the years, it turned out more and more evidence suggests both lactic acid and acidic microenvironment bodily suppress both innate and adaptive immune cells. And this is really something, and we were so excited about. So of course, there were number of mechanisms how lactic acid really induce the de-differentiation of immune cells and induce apoptosis, also through this kind of signaling pathway, also could be used as histone modification players. So I don't have to go into the detail because this is actually leading to our discovery. If lactic acid is important, if we're blocking the generation of lactic acid, that would be good. But lactic acid can be generated through the glucose pathway, also can be made by amino acid pathway. It's actually there are multiple pathway allow you to have enough lactic acid. So instead of focus on the generation, we were thinking whether or not we can blocking the lactic acid exporting process, and just blocking the backdoor. Then we're trying to find out who actually responsible for the lactic acid from the intracellular to go outside the extracellularly. It turned out MCT4 gene is a kind of related to most of the solid tumor. It's not only just a prostate cancer. So that's why we came up with the idea. So if we're blocking MCT4, so you may have direct effect that cancer cell, because they cannot get rid of lactic acid, that will leading to intracellular pH, lower pH, that could kill the cancer cell, and that's called direct effect. But more importantly, the indirect effect would be even better, because when no lactic acid or less lactic acid get out and get into the extracellular space, the pH would be increased by the system. The system will buffer in. So if the pH getting restored into the physiology level, imagine that not only just the immune cell can function better, actually all the normal cell will be happier. So that's going to get an indirect effect, most importantly, restore anti-cancer immunity. So I want to use a small video to go through this hypothesis.
Our bodies are made up of trillions of cells. Just based on the sheer quantity alone, the probability of there being mutations is likely. But when there are only 1 to a few cells that are abnormal or cancerous, our body's immune system is on standby. And in this early phase, the cancer is under control. In order to survive and grow, cancer cells generate a lot of building blocks using an altered metabolism through processes called glycolysis and fermentation. A byproduct called lactic acid is discharged out of the cell. Lactic acid pools around the cancer cells, making the microenvironment around the tumor acidic. This is one of the main methods cancer cells use to suppress anti-cancer immunity. It drastically reduces the survivability and growth of surrounding immune cells. Not only that, it also causes loss of their functions, further allowing for cancer's continual growth. The acidic microenvironment has other effects as well. It also stimulates the growth of new blood vessels through a process called angiogenesis. This promotes the spread of cancer locally and systemically. The acidity will also decrease the affinity of oxygen binding to hemoglobin, lowering oxygen levels in the area. This results in a hypoxic environment, a hallmark of aggressive cancers. So what's the most effective way to target these fundamental characteristics of cancer biology? The strategy of LAST is to develop a small molecule inhibitor that blocks the lactic acid transporter called MCT4. Successful inhibition will result in 2 main consequences. Firstly, cancer cells cannot get rid of their lactic acid byproduct. The accumulation of acid within the cell would slow down glycolysis, prevent cell growth, and directly kill the cell. Indirectly, as the acidity in the surrounding environment reduces to almost normal levels, lactic acid's effects on hypoxia and angiogenesis will begin to revert. Most importantly, those anti-cancer immune functions that were inhibited earlier will be regained. Immune cells can start attacking cancer cells efficiently again. Thus, MCT4 inhibition via LAST will not only kill cancer cells, but also restore physiological conditions to recover anti-cancer immune function. This could be one of the most powerful immune therapies that's widely applicable to most human cancers. Our bodies are made.
So I love the video because that's made by my daughter. She can speak much better than I do. So and along that line,
is your daughter for hire? That's a wonderful way of describing the complex hypotheses that you've been pursuing. So I'm sure that you have lots of colleagues that would love her to come in and narrate and draw their science as well.
Oh yeah. She actually from time to time she come to the lab and help people, and so she actually was very interested in this project herself. Okay, thank you. So along this line, and we're working with Art and Michael, and using in silicon modeling, and also in combination with real, kind of experimental screening. So we set up multiple kind of assays, and now we actually have a compound that was kind of. I just give you one more piece of data. This is really cool. So you see this some of our inhibitors when you're using co-culture system with NK cells. You use the compound alone, they inhibit some growth. You use the NK cell alone, they kill some cancers. But you use the combination, you almost get rid of all the cancer cells. That's once again demonstrate the enhancement of immune function. and you have a direct. So we also use a T-cell co-culture system, basically demonstrate the same thing. And so this is just quickly wrap up this session. So we have our silly simple working hypothesis, and based on the hypothesis that Stephen and others went on to have a discovery and demonstrate they are clinical relevant, the functional important, and study some mechanism. But more importantly, we license the discovery to a company, and this company start to generate revenue to support the research. This is the kind of the positive circle, and we generated in the lab. So I want to use the last few minutes, and because normally it's 45 minutes, and so I want to use the last few minutes to emphasize, something I'm truly excited about currently, the tumor dormancy. So I actually gather most of my people focus on this area because the reason for that one, I will give you some argument why we are so excited. So this is back to many years ago. Many years ago, one of my postdoc fellow, Francisco, and he was he's a great man, and he now is a professor in UK, and he's really cool. And when he was working with me, I just threw some challenge into him, and he wrote up an article like the one here, and in this paper, and he actually simply makes the observation along the clinical prostate cancer progression curves. One thing he realized that the dormancy ability is kind of reversely correlated with disease aggressiveness. In other words, the more aggressive cancer you have, the less dormancy ability they retain. So that might suggest the dormancy is is a born feature. It's not the gain additional feature, which is consistent with the mother's nature. You look around the animal, the plant, the bacteria, they all know how to get into the dormancy without need genomic alterations. So from that observation, he came up with the hypothesis to say this is tumor dormancy is purely epigenetic. It's nothing to do with the genomic alterations. So of course, I love the paper. And based on the paper, actually, I edited a book focused on tumor dormancy as well, but it turned out not too much going on. So that's actually give us evidence why we focus on tumor dormancy. This is mainly the argument we use when I convince my student to focus on something. I want to use the 3 principles when they select their project. Actually, this is something, first of all, you have to make sure this is a critical topic, not potentially generated a big impact. The dormancy actually is a critical because the dormancy is the one give you recurrence, give you metastasis, regardless how effective our current therapy is. So is that a critical? Of course it's critical. And also no effect to treatment or no way to manage them. So the second criteria we use, we don't want to go after so many big gurus. And because if something hot and you go after those hot area, you only go after the tail, and it was really hard for you to lead the way. So we wanted to make sure the area you get in is not that hard and not that hot, but instead it's still cold. And the cold may be also very hard to study. And then that comes to the third criteria we use. So if the area is a critical, it's everybody know that, but it's hard to everybody. How you can do better job? So that's actually you have to ask yourself, when you want to come into an area, do you have some reason to be unique and also a chance to be better? So that actually is a very important measurement. So for dormancy, we are indeed in this unique position because we are the one, we have more than 30 and hormonal naive prostate cancer model. They all respond to androgen ablation, and many of them respond to docetaxel and other known FDA approved therapy. So then in that case, we can actually induce either tumor mass dormancy, a solid cellular dormancy, all the tumor never really show the relapse in the mouse lifetime. So that's actually give us an opportunity to differentiate, what kind of dormancy gave you recurrence, give you early recurrence, or give you recurring to NEPC, or give you recurrence to AR positive CRPC. So this is really cool. So we really published this paper just early this year. And so we finally, this really come to the vision I want to share with you. So sometime people mention you want to generate a world, free of cancer, which is a good wish, but it's very hard to really reach. But if we could really manage the cancer, we know how to manage the proliferative cancer because vast majority therapeutic really target on proliferative cancer successfully. If we really know how to, meanwhile, if we know how to manage your cancer dormancy, and such as figure out what are the dormancy capable cell who really gave you dormancy, and then you can kill them all, prevent them to get into the dormancy. Or, once they get the dormancy, you can, have a good management and prevent them from recurring when even they get recurrence. So if you can target some early kind of, lineage specific commitment, transfer factor or other critical player, you probably stop their, from a very aggressive, deadly disease. So if you can do that, you can delay prostate cancer progression by another 5 or 10 years. Then we just don't have the worry, and even we have this disease. So that's actually, I was thinking this world free of the fear of cancer, or cancer fear free world, and actually can be achieved in due time. So this is the summary slide. And one thing I did not write here. I think that I really feel very thankful for the teamwork platform that Martin Gleave set up. And I think, each of us can do something, but as a team, you can be so benefited from other people's expertise. So, and also I want to emphasize, the model, and we established many of them not only unique, is that they are really truly valuable, hard to establish, but truly, give you unique, time window to learn and new things. And so the second point I want to say, and is that attractive to focus on something earlier or critically important during cancer progression, which I believe so. If you put all the money on the deadly disease space, you make people rich, you make people famous, but you don't really generate a big impact for patient, overall survive. So you got to move the landscape, to the front line. So, it's if you cannot prevent cancer from happening, at least you're trying to prevent cancer from progression. So finally, I want to share discuss these ideas. Is that realistic to realize a vision cancer-free world? And that I have my opinion. And so finally, I want to thank all the people who contribute to the work. And so the lab member is, Stephen Chow, with me for 11 years. Now he actually is a postdoc in the lab. And Cindy was a surgical pathologist, and was really smart young lady. And Connie was an immunologist who actually involved with, really dominating cancer survival mechanism, and also the MCT4 project. Dehm was also a pathologist. He actually was my first PhD student, and he actually is currently a scientist at the BC Cancer. So Jennifer is one of the best molecular biologists, and we responsible for most of the animal models. Fiona was a really, really smart lady. And I have a number of former members who contribute greatly to the science I talk about. I have many collaborators, and which are really lovely. I'm also going to collaborate with Scott, and under his NIH grant, and I believe he will be funded. I'm so excited to be part of Scott's team. And of course, and the funding is very important, and thanks all the funding agency. And again, thanks Scott, thanks everybody else for spending your valuable time listening to me. I may missing some of those, but I'm happy to answer any question you might have.
Thanks so much, YZ. That was outstanding. And you touched on, and I didn't provide any coaching, everybody, but YZ, you touched on so many topics of interest to members of our department as well. I'm sure that there will be lots of questions. I was going to lead things off, but I see Dr. Pennell has his hand up, so I'll let him go and I'll just moderate.
Thanks. Yeah, that was outstanding. I really, really appreciated your insights. I have a question about what you think drives tumor dormancy. Do you think it's an active process based on changes in the microenvironment due to, let's say, therapy, or is it a passive process where therapy selects against those proliferating cells and there's a small subset that are already dormant that then persist?
So I think I more prefer to this. I think evolution and the selection process both going on in this process. In prostate cancer field, we know when you have the stress, for example, ADT, you of course you have direct effect on cancer cell. You also have direct effect on the tumor microenvironment. So that's no doubt about that. So this on cancer cell, you're going to directly kill many of those androgen-dependent cancer cell. And then those androgen-independent cell, they not just simply sitting there, they actually undergoing de-differentiation, then preparing them for this kind of undifferentiation status. So those dormancy capable cell actually also under this transdifferentiation, de-differentiation process. So they not just stay where they were, they also learn how to survive. That's one of the interesting things I want to focus on. What are the dormant cancer cell? How much, kind of change they have? This kind of change and probably come from the stress, come from a direct effect, also the microenvironment stress. So this perhaps very similar to other dormant phenotype as well. So you have, when you have stress, that's stress all around you, direct get into you or indirect get into you, you have to receive it. And then that's the kind of a message. It's very clear for them to escape from the targeting. So I think that has to be the case. And so whether or not you have a selection going on, I believe so. And why majority of the cancer cell cannot, they have to die. And the only small proportion, they actually have this flexible ability. So that's actually what's come to our early hypothesis. We believe, the more flexible cells probably have most, kind of intact machinery associated with them. And those other cells are so stubborn, they probably have a significant damage in their machinery. They couldn't adapt themselves so well. So they only rely on something. They only rely on something. So they don't know how to change. So those are the cells actually the cells they have to die.
All right. Thanks very much.
Yep. Thank you. Enjoyed your talk. My question is, is there an effect of MCT4 inhibitor on normal cells?
Yeah, yes. So, yes. So actually very interesting. And this MCT4 and in what's the majority normal cell are not that overexpressed, and mainly in muscle cells, smooth muscles, and others. And what's the majority normal tissue don't really rely on MCT4. So MCT4 overwhelmingly, higher expressed in cancer cell. So that's where I think MCT4 knocking down mice also survive. So that's actually gave us a little bit more confidence that this toxicity issue may not be that significant, actually.
Yeah, thank you.
Wise, you had a question about the tremendous patience that your lab has when you take a surgical specimen and implant it into the renal capsule. You said for some of your PDXs, it can take multiple years for the line to establish. So what are the, and does this provide any insights to tumor dormancy? So what are the characteristics of a tumor that will, make it take off quickly when renal grafting versus really make it take a long time to take off when renal grafting? And what are the characteristics of the cells that ultimately are able to take off and ultimately become the PDX line that you develop?
So we let's try, okay, Scott, that was really cool ideas. So there are two things happening. So when you graft any kind of tissue under renal capsule, so theoretically, they are treated extremely well with a rich blood fusion. So they get enough nutrition, and also get a chance to get rid of the waste. So they living probably happily. So that's why the overall survival rate is high. So as for, which one really can become rapid growing one that from a practical point of view, give you transplantable tumor line. So for those, actually, you largely depend on the nature of the cancer cell. So as a matter of fact, when you graft the tissue into immunodeficiency mice, you already actually somehow speed up their growth because you don't have immune checking point to control them. Overall, you would speed up the growth. But eventually, the mouse still have 1 year's lifespan or also. And for most of the, primary low-grade prostate cancer, the doubling time, actually somebody, I think John Isaac's group published a paper in Clinical Cancer Research in 1985. They said those kind of cancers, the doubling time is around 472 days. I don't know how accurate it was, but to give you ideas how slow they are. So I guess when we graft the hormonal naive prostate cancer, and they all can survive. Only those ones can show relative rapid growing features that get selected. So that would take you 2 or 3 years and to realize. And that's why, that's also one of the major reasons people feel that might be impossible, because they perhaps not only just a method they use may not be right. And they probably don't know the cancer biology. Accordingly, they don't wait for that long. So that's truly reflect the nature of the tumor you graft. So for me, I feel that's a good part, because you don't want to change them too much, right? You want to be patiently watching them to perform. Then you can make a real observation by studying them.
Yeah, no, that's fascinating. And like I said, it requires a tremendous amount of patience. And maybe this is why you've had the success that you have developing the PDXs from the primary prostate cancers. It might just be that people give up before letting enough time pass by. And that's why you now have this tremendous repository of tissues that everybody in the field is benefiting from. So thank you so much, YZ, for telling us all about your work. And I see we're at time, so we should probably wrap things up. I'll keep you posted on that R01, YZ. It's
congratulations. Okay,
7th percentile. We just got to wait for it to get picked up, and then we're going to let her rip.
Beautiful, beautiful. The way to go. And thanks everybody. Thanks, Scott.
All right. Bye for now. Thanks again.
Yeah. Bye-bye.